Welcome back guys. Now in this video let's discuss about muscle physiology. The muscle physiology will be dealt under two parts. This is the part 1 where we'll be discussing mainly about the basics. In the part 2 we'll see mainly how exactly the muscle contraction will happen. There is a sliding filament theory that will be discussed in the second part. But in order to understand how exactly the contraction will happen, you need to know the basics. So the basics will be dealt in this part 1. Okay, having said that, now just tell me how many types of muscles are there? There are 3 types of muscles: skeletal muscles, smooth muscles and cardiac muscles. Why a skeletal muscle? The main important one, the skeletal muscles. Why a skeletal muscle is called as a skeletal muscle? Why? Because it is attached to skeleton. Okay, smooth muscles are around the viscera and the cardiac muscle is there in the heart. So skeletal muscles are those muscles which are attached to the bones or to the skeleton via the tendons. Okay. Now, in this video, let's see how exactly, the basic points. So, muscle physiology, part 1. Okay. Now, first basic point number 1. Without a nerve innervation, okay, without an action potential from the nerve, a muscle will not contract. So if I want to contract the skeletal muscle, okay, skeletal muscles are under voluntary control. See, skeletal muscle, they are under our voluntary control. Okay, which means you can contract them on your wish. So the skeletal muscles, for example biceps, it should have a nerve innervation. Okay, it should have a nerve innervation so that it will undergo contraction. So what is that nerve which is innervating the skeletal muscles and from where it is coming? Look here. See, this is the spinal cord. Now I am showing you here the cross section of the spinal cord. Now in this cross section of the spinal cord, now you can see the central grey matter. Okay, this is the central grey matter which is in the butterfly shape, central grey matter. Now what are these horns? These are the anterior horns, everyone knows, are the ventral horns, anterior horns. Now from this anterior horns, or in the anterior horns, who are there? See, these are the cell bodies. Cell bodies of which neuron? The cell bodies of the motor neurons. See, from here the motor neurons, especially alpha motor neuron, is coming out. Now this alpha motor neuron, okay, now this is the alpha motor neuron. It is going to where? It is going to the muscles. So muscles will undergo contraction. So now tell me, muscles are innervated by alpha motor neurons. From where these alpha motor neurons are coming? Alpha motor neurons, they are originating from the ventral horn of the spinal cord. Okay, so whenever there is an action potential which is coming along the alpha motor neuron, these alpha motor neurons will cause the depolarization in the muscles. So the depolarization in the skeletal muscle is the one which causes the contraction. Okay, now let me try to integrate here. See, for example, here in this alpha motor neuron, in the area of the alpha motor neurons, there is the anterior horns, there is a damage. Okay, there is a virus which is coming and sitting in the anterior horns. So now what will happen? So there is a damage to the alpha motor neurons. Whenever the alpha motor neuron is damaged, do you think muscle contraction will happen? No, muscles will not contract now. Okay, so what is that virus? So that virus is the polio virus. Polio virus. How it will enter into your body? Especially in children, this polio virus is going to affect mainly children, right? Polio virus enters via the fecal-oral route. What it will do? This polio virus is going to affect anterior horns of the spinal cord. Now when the anterior horns are affected, the alpha motor neurons, the cell bodies of the alpha motor neurons is going to be affected, damaged. So that do you think alpha motor neurons are going to work now? There is lesion. Alpha motor neurons are going to be damaged. These alpha motor neurons are nothing but the lower motor neurons. Remember, they are the lower motor neurons. Alpha motor neurons are the lower motor neurons. Okay. So the lower motor neurons are damaged. So muscles cannot contract now. So that will cause paralysis. So the basic point which I want to put into your mind is, the skeletal muscles, yes, they are under your voluntary control. Who is the nerve? Which is the nerve controlling this skeletal muscles? Alpha motor neurons. Skeletal muscles are innervated by the alpha motor neurons. Okay. Now, let's discuss about the structure of a muscle. See, here I am showing you a muscle. Now, inside the muscle, what do we have? We are having a fascicle. Okay. Now, this is the entire muscle. Okay, I am just showing you the entire muscle, and if you have a cross section. Now, inside the muscle, there are many subunits present. What are they? They are called as the fascicles. Okay. Now, even inside the fascicle, look, even inside the fascicle, there are many cells present. These are actually the cells. Inside a fascicle, there are many cells present. These cells are called as the muscle fibers. So, muscle fibers are the real cells. They are the cells of the muscle, the cells of a muscle. Inside a muscle, there are many muscle fibers. So, these muscle fibers are the real cells. Now, for example, imagine this is one muscle fiber. This is the one which contains actin and myosin. Inside this muscle fiber, actin filaments and myosin filaments are present, which are helping in the contraction of the muscle. So, inside a muscle fiber, see, there are myofibrils. Okay, inside the muscle fiber, there are myofibrils. This is a muscle fiber. Okay, this is a cell. Inside the cells, myofibrils are present. These myofibrils are nothing but the actin and myosin. Okay, now for example, for your understanding purpose, let me show you something like this. See, this is the muscle fiber. Okay, now inside this muscle fiber, what do we have? We have actin filaments and myosin filaments. Okay, actin and myosin, something like this. Okay, now tell me, muscle, inside the muscle we have many fascicles. Inside a fascicle we have muscle fibers. Muscle fibers are the real muscle cells. They are the units of a muscle. Now, inside a muscle fiber, we have actin and myosin filaments. They are called as myofibrils. Okay, they are called as a myofibril. Okay. Now, let's begin something like this. Let me draw one muscle fiber for you. Okay. Look here. Now I am talking about the skeletal muscle. Inside the skeletal muscle, I am talking about the muscle fiber. Okay. Now, this is the cell membrane. Each muscle fiber is a cell, right? I have already taught you. So, these muscle fibers, they are also surrounded by a cell membrane. So, this cell membrane is called as sarcolemma. Okay. Sarcolemma. Now, look, this sarcolemma, it is having some invaginations like this into the cell. The cell membrane, normally cell membrane is straight like this, right? Now, the sarcolemma is having a deep invagination like this, and it's again coming out. So, this invagination which I am showing here is called as T-tubule. Okay, it's called as T-tubule. Okay, so many such T-tubules will be there. Okay, here like this again. So, this is one muscle fiber which I am drawing here. Okay. Now, see, inside the skeletal muscle, let me show with the pink color. There is T-tubule. Okay. Have you ever heard about L-tubule? T-tubule and L-tubule. So, T-tubule, you know, it's a part of the sarcolemma. Okay, it's a part of the sarcolemma where the sarcolemma is invaginating into the cell. That's a T-tubule. Now, look here. Inside this muscle fiber, there is a structure present with the two globular ends like this. Okay, with the two globular ends. They are present longitudinally. Okay, so these are called as L-tubules. Okay, this pink color one, These are called as L-tubule. One is a transverse, so that's called as a T-tubule, and other is a longitudinal, which is present in the sarcoplasm. Within the cell, this is present. This is called as a L-tubule. Okay. See, T-tubule, you know, it's a part of Sarcolemma. But then what is this L-tubule? This L-tubule is nothing but Sarcoplasmic reticulum. Okay. This is nothing but the endoplasmic reticulum within the muscle cells. So this is nothing but Sarcoplasmic reticulum. Okay, Sarcoplasmic reticulum. See, this Sarcoplasmic reticulum, at their ends, they are having these globular portions. Okay, the globular portions. These globular portions, they are called as cisterna. Okay, so they are called as a terminal cisterns. Okay, cisterna. Now, what are the important points which you need to know, especially for your exams? They will ask you. This Sarcoplasmic reticulum, it contains what? What does it contain? It contains lots and lots of, tons and tons of calcium. Okay, so calcium is going to be present inside. So Sarcoplasmic reticulum is the storehouse of calcium. Especially the calcium is going to be concentrated, okay, in the cisterna region. So let me write here, Sarcoplasmic reticulum, it's a storehouse of calcium. Where calcium is going to be stored, especially calcium is concentrated in cisterna region. Okay, done. Now, what else you should know? See, I have already taught you these muscles. They are innervated. This is a muscle fiber, no? It's a muscle cell. So it should have an innervation. There should be some neuron, the alpha motor neuron, it should come and it should innervate this muscle fiber. Okay, so I am showing that alpha motor neuron. See, this is the alpha motor neuron. It is coming out and it is innervating. It is giving the innervation to the muscle fiber. See, now what it will do? It is coming, it is getting depolarized. It will give the the neurotransmitter. The neurotransmitters will be released here. We will discuss about that. So this is the alpha motor neuron here. I am discussing about the alpha motor neuron. Now, before going any further, I want you to know about the sarcolemma, some important points about the sarcolemma. See, in the sarcolemma, that is the cell membrane of the muscle. In the sarcolemma, there are certain proteins present. Okay, so what are these proteins? The proteins are mainly of two types. They are peripheral proteins as well as the integral proteins. Okay, there are peripheral proteins and integral proteins. You know, in general physiology also we have discussed what is a peripheral protein, which is present either on outside or inside. Okay, peripheral. Integral protein means they are present throughout. Okay, they are present throughout the cell membrane. Okay, penetrating through the cell membrane. So there are peripheral proteins present as well as integral proteins present. What are the examples of integral proteins? Examples of the integral proteins include dystroglycan, okay, dystroglycan and sarcoglycan. Okay, dystroglycan and sarcoglycan. What are the examples of peripheral proteins? Peripheral proteins include dystrophin, okay, dystrophin as well as syntrophin. Okay, so dystrophins and syntrophins. Now, it is a complex. All these are the proteins which are present within the cell membrane. Okay, they are present within the cell membrane. And they are the ones which maintain the strength, the tensile strength of the cell membrane. Now, let me show you the image. See, here you are concentrating on a cell membrane. Now, in the cell membrane, I have already taught you two integral proteins and two peripheral proteins. What are the integral proteins? See, the first integral protein which I am discussing is called as the integral one, a dystroglycan. Okay, dystroglycan, integral protein. See, this dystroglycan is having alpha subunit, beta subunit. Okay, so let me draw a simple image for your understanding. Look here, this is the cell membrane. Okay, in the cell membrane, what do we have? We have this dystroglycan. Okay, alpha subunit as well as the beta subunit. Dystroglycans are there. Now, one more integral protein is called as sarcoglycan. So here is the sarcoglycan which is having many subunits. Okay, sarcoglycans. Let me show you the sarcoglycan here in this image. See, these are the sarcoglycans. Alpha, beta, gamma, delta. So what are these? These are sarcoglycans. And one more protein is there which is called as a sarcospan. Okay, sarcospan is also an integral protein. Okay, sarcospan is also there. If you want to take it, you can take it here. Okay, let me write sarcospan. Okay, so dystroglycans, sarcoglycan, sarcospan. What are the two peripheral proteins which I have discussed? The two peripheral proteins which I have discussed are the dystrophin, syntrophin. Let me show you the dystrophin and syntrophin here. See, this is a peripheral protein. So it is present either outside or inside. So now I am highlighting this. Look, this one which I am showing you here, which I am highlighting, this pink color one. See, this is the dystrophin. Okay, all this is the dystrophin. Okay, so okay, let me show you the green color. See, this one which I am highlighting. So it is present internally in the cell. So this area, entire thing is called as a dystrophin. So dystrophin, okay. Along with the dystrophin, what else is there? Dystrophin as well as syntrophins. So here look, syntrophins. Here are the syntrophins present. So dystrophins and syntrophins, they are present inside the cell. Okay. Now this dystrophin, it is attached to what? Dystrophin, it is attached to actin filaments. Okay, this actin is a part of cytoskeleton. Okay, this actin is not, I am talking about the actin myosin, this is a different. Okay, so this actin, look here, this dystrophin, it is attached to actin filament, cytoskeletal actin, which will give the shape and structure of the cell. So now, simple, look here, alpha and beta, what are they? These are dystroglycans. Okay, these are the dystroglycans. Now what is this green color fellow? Alpha subunit, beta, gamma, delta, what are these? Now this fellow is a sarcoglycan. And along with them one more thing will be there. Okay, let me show you the red color here. There is also something called as a sarcospan. Okay, so these three are the integral proteins. Okay, now inside the cell, I am showing with the black color. Inside the cell, there is something called as a dystrophin. Okay, this is dystrophin. This peripheral protein is called as a dystrophin. Now, this dystrophin is also attached to syntrophin. Okay. Now this dystrophin internally inside the cell, it is attached to what? It is attached to the actin molecule. Okay, filamentous actin. So dystrophin, it is attached to actin inside the cell. Okay. Now this dystroglycans externally outside the cell, outside the muscle fiber, what they are attached to? See, they are attached to, okay, outside they are attached to other filaments like lamin. Okay, now let me show you the image. Look here. See, this is dystroglycans. Outside they are attached to what? They are attached to laminin. Okay, see, this is the laminin. Okay, extracellularly it is attached to laminin. Done. So now tell me, in the sarcolemma, we are starting with the sarcolemma. In the sarcolemma, there are different types of proteins present. Integral proteins as well as peripheral proteins. Integral proteins include dystroglycan, sarcoglycan, sarcospan. Externally, these integral proteins are attached to, especially dystroglycan, it is attached to laminin. Internally, these integral proteins are attached to dystrophin. This dystrophin in turn attached to the actin. Now, this whole complex, this whole thing, okay, is giving what? The whole thing gives the tensile strength to the muscle fiber. It gives the tensile strength to the muscle fiber as well as the sarcolemma. Okay. Now, what's the problem? The problems here are whenever there is any damage to these proteins, or any problem with these proteins, or absence of these proteins, mutation of these proteins, then problems will occur. So let me start from here. Absence. Okay. So the dystrophin gene, it is mutated, the largest gene in the human genome, the dystrophin gene, it is mutated, and there is absence of dystrophin. Okay, the dystrophin is absent. When the dystrophin is absent, it is going to cause Duchenne's, okay, muscular dystrophy. Okay, so Duchenne's muscular dystrophy is due to complete absence, okay, complete absence of the dystrophin. For example, if dystrophin is not enough, not sufficient, then it is going to cause, okay, something called as Becker's muscular dystrophy. Okay, so dystrophin, it's now not good, deficient. It's going to cause Becker's muscular dystrophy, which is a milder variant. This is more severe. Duchenne's muscular dystrophy, DMD, Duchenne's muscular dystrophy is due to complete absence, but defect or deficiency will lead to the Becker's muscular dystrophy, which is a milder form. Many questions were previously asked in the exam from this topic, Duchenne's muscular dystrophy. If a patient is having Duchenne's muscular dystrophy, imagine there is this child where this dystrophin is not present, so the muscles are weak, there will be muscle weakness. So there will be muscular weakness, okay, muscular weakness will be seen from childhood onwards. So when you ask this person to squat, okay, sit down, and if you ask this person to stand, because of the muscle weakness, he cannot stand just like a normal person. He uses his upper extremities like this to stand up his body, to lift his body. See, the sign is called as Gover sign, using the upper extremity, like upper extremities to lift his body. Okay, so this is called as Gover sign. Gover's sign. It is going to be positive. Gover sign is positive. Next, what? The muscles are undergoing damage. The muscles will undergo damage. Because of that, especially in the calf muscles of this particular patient will undergo atrophy. The calf muscles will undergo atrophy. In the place of calf muscles, fat will start to deposit. So now if you look at the calf muscles, they will look like big calf muscles, but actually they are not calf muscles. It's nothing but the fat accumulation. So this is called a pseudo hypertrophy of the calf muscles. Okay, not the real hypertrophy, pseudo hypertrophy. So there will be pseudo hypertrophy of calf muscles. Okay, pseudo hypertrophy of the calf muscles. So these are the important points. Deficiency of dystrophin will cause Becker's muscular dystrophy. Complete absence will cause Duchenne's muscular dystrophy. And in Duchenne's muscular dystrophy, the patient is going to have Gover sign positive. Muscular weakness will be there. Because of that, when he tries to stand his, lift his body up, he will use upper extremities. So that is a Gover sign positive, and pseudo hypertrophy of the calf will be seen. And these patients, muscles are very, very weak. Even for the breathing also, the skeletal muscle, right? Diaphragm is a skeletal muscle. So the skeletal muscle is going to be weak. So these patients will die. Okay. So death will occur due to respiratory distress. Okay. Important MCQ. Okay, respiratory distress can lead to the death. Now, we have also studied about the sarcoglycan complex. Here, these guys. Okay, alpha, beta, gamma, delta, sarcoglycans. So, whenever these sarcoglycans, whenever they are affected, that's going to cause which disease? That's going to cause limb girdle, myopathy. Okay, so limb girdle myopathy or limb girdle muscular dystrophy is going to be seen whenever there is defect. Whenever there is defect in the sarcoglycans, okay, defect in the sarcoglycans will cause limb girdle muscular dystrophy or limb girdle myopathy. Okay. So with this, the sarcolemma important points are completed. Now again, look here. Here I am talking about the sarcolemma. What is this invagination of the sarcolemma into the cell? This invagination, this is nothing but the T-tubule. Now look again one more T-tubule. T for transverse. Okay, transversely they are coming into the cell. T-tubules. Now see, on this T-tubule, okay, on this T-tubule, there is a calcium channel present. Okay, there is a calcium channel present. This calcium channel which I am drawing over here, this is called as DHPR, dihydropyridine receptor calcium channel. Okay, dihydropyridine receptor calcium channel. Okay, which is also called as CAV 1.1, voltage gated calcium channel 1.1 subtype. Okay, so now tell me, on the T-tubules, which type of calcium channels are present? Dihydropyridine receptor calcium channel is present, CAV 1.1. Okay, now look here. What is this? This is the sarcoplasmic reticulum. Okay, sarcoplasmic reticulum. Now what are these terminal regions are called as cisterna, where maximum calcium is going to be stored. Now on this, one more calcium channel is present. Okay, one more calcium channel is present. What is this calcium channel? This calcium channel is called as a ryanodine receptor. Okay, subtype 1. So ryanodine receptor type 1, it is present on the sarcoplasmic reticulum. Sarcoplasmic reticulum. Okay. Now why do we need to know all this? Is because, see, whenever the muscle, the alpha motor neuron is firing, okay, the alpha motor neuron is firing. Okay, let me draw here the alpha motor neuron. This is the alpha motor neuron. Now it is firing. Now this alpha motor neuron, what it will release? You should know. This alpha motor neuron, it will release acetylcholine into the synapse. This synapse, now here it is called as a neuromuscular junction. This is the neuromuscular junction, right? The neuron and the muscle, there is a junction, neuromuscular junction. Now this acetylcholine, it is going to act on its receptor. There is a receptor for acetylcholine. That is the nicotinic M receptor, NM type of receptor. Okay, it's a NM type, nicotinic M, nicotinic receptor which is present in the muscle, in the neuromuscular junction. So this acetylcholine, it will come, it will bind here. Now what happens? Now the muscle membrane, it will start to depolarize. Okay, now the muscle membrane is going to activate. There is influx of sodium. Sodium will influx. Okay, because when the acetylcholine comes and binds on the nicotinic receptors are nothing but the channels. They are the ion channels. They will be opened and sodium will start to come into the cell. Now what the sodium will do? The sodium will move to the surrounding areas. The sodium will move into the surrounding areas, causes the depolarization, subsequent depolarization. So now what happens again? New sodium channels will start to open and sodium will start to come into the cell. Now this sodium will go into the surrounding areas, causing the depolarization. So there is a depolarization in the membrane, in the muscle membrane or the sarcolemma. Now you should know what this depolarization will do. Now this depolarization, it spreads through the T-tubule also, it comes through the T-tubule. Now when it is coming through the T-tubule, what it will do? It will activate which channel? It will activate dihydropyridine receptor calcium channel. So this DHPR, dihydropyridine receptor calcium channel is going to be activated. Now this DHPR, what it will do further? Now this DHPR, it will, whenever it is activated, mechanically, see, now this dihydropyridine calcium channel, now one of its subunits, mechanically it interacts with ryanodine receptor. Okay, mechanical interaction will happen. Okay, why? Because first depolarization spreading along the T-tubule like this. Now this depolarization is going to first activate the DHPR channels. Now the DHPR channels, mechanically they will go and interact with the ryanodine receptor. Now do you know what happened to the ryanodine receptor? So far the ryanodine receptors are closed. Because of this coupling, because of this mechanical interaction, it will start to open. Now this ryanodine receptor channels, they are opened. Once they are opened, what will happen? All the calcium, whatever is there in the sarcoplasmic reticulum, this calcium will start to leak out. So this calcium is coming out to the sarcoplasm. Now for the first time, the calcium is going to leak out of the sarcoplasmic reticulum. Now what this calcium will do? the calcium. In the next video we'll discuss the calcium will go and bind with something called as a troponin. So troponin and a tropomyosin will move away from the active sites. So at the end of the day, because of this calcium only, the muscle contraction will happen. Muscle contraction. Okay, muscle contraction will happen. Okay, muscle contraction will happen. So now tell me, here first alpha motor neuron is depolarized. There is a depolarization of the alpha motor neuron. Okay. Now this, because of this depolarization of the alpha motor neuron, first it is going to release the acetylcholine. First alpha motor neuron is activated. Action potential is spreading along the alpha motor neuron. It releases the acetylcholine. Acetylcholine binds with the nicotinic receptors in the neuromuscular junction. Nicotinic receptor is activated so that it allows the sodium into the muscle fibers or muscle cells. Now that will cause a depolarization. See, the entire depolarization is spreading. The depolarization is spreading along the T-tubule. Now on the T-tubule, the DHPR channels are going to be activated. Now this DHPR channels, mechanically they will cause interaction with the ryanodine receptors. Now when the ryanodine receptors are opened, the calcium which is stored in the sarcoplasmic reticulum, sarcoendoplasmic reticulum, is going to leak out into the cytoplasm, will cause a contraction of the muscles. Okay. So this is what happens in the skeletal muscle. Okay. This is what happens in the skeletal muscle. Now I just want to make one more diagram to actually tell you a difference, a little differences between a skeletal muscle as well as a myocardium. Okay, the myocytes. Now I am dividing the page into two halves. For example, look. Okay, I am dividing the page into two halves. On one side, okay, let me draw here. This is the sarcolemma. Now, come on guys, tell me what is this invagination? The first invagination here I am drawing, this is called as the T-tubule. These are the T-tubules. Okay. Now what is this pink color guy? This pink color guy, whatever I am drawing here, is the sarcoendoplasmic reticulum. Okay, sarcoplasmic endoplasmic reticulum, which is a storehouse of calcium. You already know it. Now, please tell me, what is this receptor which is present over here? Not a receptor actually. It's a ryanodine. Sorry, not ryanodine, dihydropyridine calcium channel. Okay, this is the DHPR. Okay, the green color guy here. This is dihydropyridine receptor. Now, who is he here? This is the RYR channel. Okay, ryanodine receptor calcium channel. Okay, now look here, in a skeletal muscle, what is anatomically different? Okay, what is that difference which is present between the skeletal muscle as well as the myocardium is same. Side to the T-tubule, on both the sides, there are this sarcoendoplasmic reticulum present. This is the sarcoendoplasmic reticulum which is present on both the sides of the T-tubule. Now, if this is the T-tubule, on one side there is one sarcoendoplasmic reticulum, on the other side there is one more sarcoendoplasmic reticulum. But look here, in myocardium, what are the differences which you should know? Even on this myocyte, the ventricular myocyte which I am drawing here, this is one T-tubule, okay. This is the other T-tubule, okay. Now, look here, there will be only one sarcoendoplasmic reticulum. Okay, only one sarcoendoplasmic reticulum is present. Let me draw a small size. This is sarcoendoplasmic reticulum. Why I am drawing here small? Because the sarcoendoplasmic reticulum, this endoplasmic reticulum which is present in the skeletal muscles is bigger. This is also a question. Okay, these are the skeletal muscles. In skeletal muscles, the sarcoendoplasmic reticulum are bigger in size. Okay, when compared to the myocardium. Now here look, in the myocardium, small sarcoendoplasmic reticulum. So are there two sarcoendoplasmic reticulums on both sides of this T-tubule? No, that's the first thing. Next, what are the other differences? Here, again, what is this channel? Can you tell me? This green color channel which is present on the T-tubule? Yes, this is also dihydropyridine receptor, DHPR, which is also called as calcium, voltage gated calcium channel 1.2. Okay, 1.2. So here this is DHPR. Okay, whatever the DHPR which I am writing over here, this is voltage gated calcium channel. CAV means voltage gated calcium channel. V stands for voltage. Voltage gated calcium channel 1.1. Okay, 1.1. Now in a myocyte it is voltage gated calcium channel 1.2. That's a one difference. And look here, what is this channel? So this channel which is present on the sarcoendoplasmic reticulum, on the sister name. You guys know what this is called as the RYR, ryanodine receptor channel. This ryanodine receptor here, it is called as a RYR2, RYR subtype 2. Okay, here it is RYR1. Okay, RYR1. Here it is RYR2. Okay, that's the one difference. So DHPR channels are different as well as the RYR channels are different. And anatomically, there is only one L-tubule side to the T-tubule. Now look here, what's the difference is, now this is the this is not the neuron. Now the action potential is coming, you know how the action potential transmits in the from the SA node to AV node, from the AV node to the bundle of His, from the bundle of His to the Purkinje fiber. Now Purkinje fiber is stimulating, okay, now Purkinje fiber is stimulating this myocyte. Our action potentials are coming from the surrounding myocardial cell via the gap junctions. Now look here, when this muscle membrane, when the sarcolemma, when it is undergoing depolarization, okay, now what happens is this depolarization is going to spread along the T-tubule. Now this depolarization is going to first activate which channels? It's going to activate the DHPR channels. Okay, now important point is, now here the DHPR channels, now they will open. It's not the mechanical interaction now. Now it's not the mechanical interaction. Now it's the DHPR channels, they will open up. Okay, they will open up. So when they open up, do you know what happens? Usually calcium is more outside the cell. Whatever the calcium which is available outside the cell, some amount of calcium, the calcium from the extracellular fluid, this calcium, it will start to enter into the cell. Okay, this calcium, see this calcium is coming from the extracellular fluid via the DHPR channels. Okay, so now what happens? DHPR channel open. Now the calcium came into the cell. Do you know what this calcium will do? Now this calcium will open, it will come, this calcium, it will come and act on the RYR channel. So now the calcium will cause opening of the RYR channel. It's not the mechanical interaction which is causing the opening of the RYR channel, but the calcium whatever came from the extracellular fluid will cause the opening of the RYR channel. So now RYR channels, now they are opened. Now they are opened broad. The calcium which came through the DHPR channel is very, very less. This calcium influx is very, very less. But this is the real calcium which will cause the opening of the RYR channels. So when the RYR channels are opened, do you know what happens? Now the calcium whatever is there inside the sarcoendoplasmic reticulum, now this calcium, it will come out. Okay, the calcium is now coming out. Okay, so what this calcium will do? This calcium will cause contraction of the myocyte, the heart muscle. Okay, contraction. So now tell me, what is the difference between a skeletal muscle and between a myocardium? So I have already shown you the differences, the two important differences, the anatomical differences. Okay, DHPR channels are different in skeletal muscle as well as the myocardium. The RYR channels are different. The T-tubule L-tubule relationship, okay, the sarcotubular, okay, the relationship is little different, and the interaction is also different. Here, because of the depolarization, see, because of the depolarization, DHPR channel is interacting with the RYR channel mechanically. So this interaction here in skeletal muscles is called as very, very important. This is called as electromechanical coupling. Okay, this is called as electromechanical coupling. Electro in the sense here the electrical activity. This electrical activity is causing the mechanical interaction between the DHPR channel as well as the ryanodine receptor. So this is called as electro. Electro stands for the this activity, electrical activity, the depolarization along the muscle membrane is causing the mechanical interaction between the DHPR as well as RYR, leading to the release of the calcium from the RYR channels. This is called as the electromechanical coupling. Okay, calcium will come out and causes the muscle contraction. Now here in myocardium, Yeah, depolarization is happening. So it's not the mechanical interaction but the chemical interaction. So this is called as electrochemical coupling. Okay, electrochemical coupling. So electrochemical coupling is seen in which cells? Skeletal muscles or cardiac myocytes? It's seen in the cardiac myocytes, where the depolarization along the muscle membrane will cause the release of a calcium. I should not release, I should say it will cause the influx of calcium via the DHPR channels. See, via the DHPR channels, first calcium will come into the myocardium. Now this calcium will cause opening of the RYR channels. So whatever the calcium that is coming out of the sarcoplasmic reticulum will cause the muscle contraction. Okay, so after this, what else you should know for your exams? Electromechanical coupling, electrochemical coupling, I have discussed. Okay, done. Now for your exam, what else you should know is, see, the RYR channel, skeletal muscle. I am talking about the skeletal muscle now. Now the RYR, that is a ranodin receptor 1. This RYR channel, if it is having gain of function, it is overactivating. Okay, gain of function. If it is over functioning, tell me what happens. Normally, whenever there is a depolarization, whenever the DHPR channel is activated, then only the RYR channel should function and release the calcium. But because of certain mutations, if it is over functioning, lots and lots of calcium will be released. So that calcium will cause the too much contraction of the muscles. Okay, so too much contraction of the muscles will cause the breakdown of ATP and release of the temperature. Okay, energy will be released, ATP will be broken down. So while the process of the ATP breakdown, when the muscles are contracting, we all know that temperature will be produced. So the point which I want you to know is, because of the mutation of this RYR channel, the patient can suffer with a condition called as malignant hyperthermia. Okay. So usually this malignant hyperthermia is going to be precipitated. It's going to be precipitated by inhalational anesthetics. Okay, inhalational anesthetics like halothane, desflurane, or certain depolarizing muscle relaxants like succinylcholine. Okay, see, these are the drugs. For example, let's take them as a drugs. Now, in a patient who is having this mutation, okay, this ranodin receptor type 1 channel, it is mutated. Now, in that particular patient, whenever you use this inhalational anesthetic agents or this muscle relaxant, succinylcholine, it's a muscle relaxant. Whenever you use, immediately the patient will go into a condition called as malignant hyperthermia. Okay, now what you need to do? For example, imagine I am the person who is having malignant hyperthermia. What you need to do? You need to block which channels? You need to block the RYR channels. Okay, RYR channel is a problem, right? It is continuously releasing the calcium. So RYR channel need to be blocked, and the drug which is used is dantrolene. Okay, dantrolene sodium. So dantrolene sodium is a ranodin receptor channel blocker. Okay, which is used in the treatment of malignant hyperthermia. Okay, this is one integration which you need to know. Apart from this, what else you should know? See, this DHPR, RYR channel completed, integration completed. DHPR channel is also called as CAV 1.1. Okay, CAV 1.1. Now, if this channel, if it is mutated, it will cause which condition? It is going to cause a condition called as hypokalemic periodic paralysis. Okay, hypokalemic periodic paralysis is going to be seen. Okay. Next, what else you should know is muscle relaxation. Okay, integrations are completed. Muscle relaxation. See, calcium is the one which causes the muscle contraction. How exactly it causes the muscle contraction, later we will discuss. Now, during muscle relaxation, what should happen? Think logically and tell me. See, this is the sarcolemma, you know. Now the sarcolemma, what it is having? These are the T-tubules. Okay. Now, what is this one? This is the sarcoendoplasmic reticulum with the cisterna, terminal cisterna. Okay. So, already calcium came into the cell. Okay. Now, this calcium, it have to go back. It have to be again sequestered back into the sarcoendoplasmic reticulum during relaxation. Then only muscle relaxation will happen. So, which transporter? There is a transporter here on the cell surface. There are certain transporters which will cause the return of the calcium back into the sarcoendoplasmic reticulum. So, all this calcium will be returned back. So, now already you guys know inside the sarcoendoplasmic reticulum tons and tons of calcium is there. Here lots and lots of calcium is there. So, now tell me, now you are moving this calcium according to the concentration gradient or against the concentration gradient? You are moving it against the concentration gradient. Already in the sarcoendoplasmic reticulum loads and loads of calcium is there. Only little calcium came out during the muscle contraction. So, all this calcium need to be sequestrated back from low concentration to high concentration. So, definitely we are going to use active transport or passive transport? We are going to use active transport. Okay, we are going to use active transport. See, this active transport which I am discussing now, okay, see, it is called as, this transport, the name of the transport is called as SERCS, SERCA. So, SERCA stands for Sarcoplasmic, Endoplasmic, Reticulum, calcium ATPase. Okay. So, sarcoplasmic, endoplasmic reticulum, calcium ATPase. Okay, it's an example of primary active transport. By using ATP, calcium will be sequestered. Okay. Now, important point is SERCA. Okay, when it is damaged or the gene for the SERCA, when it is mutated, it is going to cause myopathies which are called as Brody's myopathies. So, SERCA gene mutation. There is a gene for SERCA which is called as ATP 2A1 gene something. So, if the SERCA, if it is damaged, if this protein is damaged, then it is going to cause Brody's myopathy. Okay. So, that's the MCQ. So, now tell me, what are the important points which you need to know? Muscle innervated by alpha motor neuron. These alpha motor neurons are going to release acetylcholine. That acetylcholine will cause the depolarization of the muscle membrane. The muscle membrane is having invaginations called as the T-tubules. On the T-tubules, there are channels called as a DHPR channel or calcium, voltage gated calcium channel 1.1. Now, inside the cell, what do we have? We are having a sarcoendoplasmic reticulum which is called as the L-tubule with the 2 terminal cisterns, inside which lots and lots of calcium is stored. Now, when a muscle membrane is depolarized, DHPR channel, I am talking about the skeletal muscle now, okay, the DHPR channel is going to be activated. The DHPR channel interacts with the RYR channel mechanically, so the RYR channel will open and releases the calcium, okay, releases the calcium. The calcium will help in the contraction, done. After the contraction, during relaxation phase, what will happen? The calcium will go back into the sarcoendoplasmic reticulum with the help of calcium, sarcoendoplasmic reticulum, calcium ATPase, which is called as SERCA. If it is mutated, if it is damaged, if that protein is damaged, it is going to cause Brody's myopathy. What is the difference in myocardium, in heart, in myocardials, in the cardiac muscle? The difference is RYR channel is different, DHPR channel is different, and surrounding the T-tubules, 2 sarcoendoplasmic reticulums are not there, but only 1 sarcoendoplasmic reticulum is there. Okay. Next, what is the difference? In skeletal muscle, there is electromechanical coupling is seen, but in muscle cells, in cardiac muscle, there is electrochemical coupling is seen. Next, in skeletal muscle, look here, in skeletal muscle, okay, let me show you here, this one. In skeletal muscle, there is only one source of calcium for the contraction. See, muscle membrane is depolarized. Okay, muscle membrane is depolarized. DHPR channel opens the RYR channel. So RYR channel is going to release the calcium. So this calcium is helping in the contraction. That's it. But it's a one source of the calcium. But here in myocardium, little difference is, see, calcium is coming from the extracellular fluid, one thing, and also calcium is coming from the sarcoendoplasmic reticulum. There are two sources of calcium. But what is the important calcium? It's the first calcium. See, this first calcium, whatever is coming from the extracellular fluid, this is called as the essential calcium. Okay? This is the essential calcium, which will open, okay, this calcium, whatever is coming from the outside, it will open the RYR channel. Okay, it will open the RYR channel. So RYR channel opens, calcium comes out and causes the contraction of the myocytes. Okay, so with this, the basics, the foundation for the muscle contraction or the muscle physiology is completed. In the next video, we will exactly see how the sliding filament theory happens. The actin, myosin, all the important points will be discussed in the next video. Hope the video is helpful. Thank you.